Great. So it is my pleasure to introduce José Otero, who's an MD, PhD, neuropathologist and associate professor of pathology at Ohio State. Jose did his pathology residency and neuropathology fellowship at UCSF, which is a powerhouse for neuro-oncology research. And his research interests span stem cell biology and also molecular diagnostics in diffuse gliomas. And so today, Jose is going to be talking to us about providing molecular insight for resource-strained patients with machine learning-based workflows. Thank you.
Okay, thank you for that. So I'm going to try to see here and go to presenter mode. So can you all see the screen?
Yes.
Yep. Okay.
That's good.
Well, thank you for giving me the opportunity to share with you some of the work that we're doing here at Ohio State in our lab. You know, I'm just a little background. I'm not really, I've never gotten any formal training in data analytics. I just had to learn to deal with some of the challenges that we've had over the years as our science has moved in this direction. There's some interesting things that I think we have to talk about as we're trying to implement these types of computational approaches. You know, one issue to think about is that we have to be a little bit grounded. Here is a graph from a paper in the Lancet that discusses the distribution of pathologists worldwide. And so to put things in perspective, every single person on Earth who undergoes an oncological treatment must have had a pathologist diagnose a cancer. That's an absolutely important part for the workflow. Despite that requirement, pathology is routinely understaffed and highly strained. Here in the green, for example, in the United States and Canada, there's one pathologist per 40,000. But in some countries, such as in Southeast Asia or in Africa, we're talking about one pathologist per 5 million. So that's really difficult challenge for global medicine. Pathology is also a highly complex integrated specialty. So there's not just one person looking at things on a slide. That person's integrating data from lab information, histology, immunohistochemistry, cytopathology, you name it. And it requires an integrated expertise in infrastructure. And even more important than that physical infrastructure is a human infrastructure, which is lacking in most places. There's lacks of training, lack of equipment, standard operating procedures are not in place. Quality control and quality assurance issues as well abound globally and even in the United States. So here, for example, is a picture at Ohio State of something called the gross room. For those of you who aren't pathologists, that's where, for example, if a patient's tissue is resected out, it's brought in and there's a dissection. The whole process of generating the material for diagnostics begins here. So this is what it looks like, right? And it's actually very clean. This isn't just the first day, that actually is like clean like this at Ohio State. So these are 2 stations, for example, there's a computer here, everything's at the person's fingertips and so forth. This is a picture of a gross room in a country that actually by World Bank criteria meets a developed economy, right? So we're not talking about a poor country like Tanzania or Suriname. We're talking about a country that has just an understaffed university hospital. And yes, this is mold growing on the wall. So when you're talking about machine learning and AI, you know, maybe it's not impossible to train an algorithm here that generalizes to something like this, but maybe we can all agree that it would be very challenging, okay? A further burden here is the fact that cancer, as the population is living longer, patients are, more cancers are occurring. Here is the growth of the US population, for instance, and you can see the increase in cancers, the incidence is increasing. And if you plot by change, you can see how much more rapidly and disproportionate the increase in cases is happening. And we're estimated within 20 years to potentially have a 20 to 40% increase in the number of cases of cancer. So this further burdens a national and global pathology workforce. Another point also to consider is that as we're understanding more of the molecular insights into therapeutics, it's much more, each diagnostic entity becomes much more complicated to understand. And today I'm going to talk about brain cancers, but many of the problems that are occurring, or challenges rather, that occur in neuro-oncology are quite translatable to other areas. So one of the trends that are emerging in brain cancer diagnostics is the focus on molecular patterns. So in 2016, the World Health Organization generated a classification system. This is a page right here out of the WHO. These are the different entities. Brain tumors were the first solid tumor class that required molecular characterization to elicit a diagnosis. So that's all fine and well. It's great. It's a big improvement. The problem is that these assays are not well reimbursed. And in many countries, they're absent. Forthcoming in 2021 will be an update to this classification, which not only extends the number of molecular genetic requirements, it also includes entities that require methylation profiling to diagnose. Let's put this picture here. So we still don't know how to pay for molecular genetics. Now we're having to start thinking about problems of molecular epigenetics, which are a completely different technological platform. And there's no way anyone's going to pay for this. So the problem that we've got is that the molecular genetic insights are occurring at a pace that's by far exceeding our capacity to develop a healthcare problem or a healthcare solution and policy that can finance that. And that's not just occurring in the United States. Every country is dealing with this problem. So how can we meet some of these problems? Well, I'm probably preaching to the choir, but I do feel that machine learning workflows can improve this. It might be able to fill in a gap of applying clinical diagnostic aids and help make informed decisions that might reduce redundant testing and improve your pretest probabilities for expensive assays. But what are some of these unique challenges? Well, principally is that primary brain cancers are very rare. So worldwide, the incidence ranges from 2.6 to 3.7 per 100 cases per 100,000. So that means very few data sets to train on. The other issue that happens is that right now you end up having high resolution data on each patient, right? And so you have a low number of subjects with multiple features to model. Also, not all testing is appropriate for each tumor entity. So many of these brain cancer data sets have so much missing data that they're beyond being saved by simple imputation methods. The current repositories such as the Cancer Genome Atlas and the Chinese Glioma Genome Atlas, they have some H&E stained images, which is the slide preparation, but they're generated off of flash frozen tissue. And that morphology is not translatable and not generalizable to standard workflows. So with those challenges, we started thinking, well, how can we start thinking about ways to improve things? And so we generated a thought experiment, which was, okay, well, we've got these like entities here. Each entity the scientific literature has classified it about what the demographics are, what the features are, and so forth. Let's make a simulation of this data set because we don't have the information of every single patient. Let's see if we can come up with a model. And so we did first a meta-analysis of the scientific literature pertaining to the classification of gliomas. We focused on common types of assays that would be present throughout, for example, H&E staining and things like that. And we generated this simulated brain cancer population. We call this just a simulation. We had 4 types of data sets or types of data that we simulated. Very simple clinical features such as the patient age, gender, and sight. Histological features such as things like desmoplasia, which means something to us pathologists, immunohistochemistry such as GFAP, and then molecular features such as IDH mutation, which are very important for gliomas. And we focus on a very small number of molecular features because these features are very easily reimbursed by US billing policies. So we basically just used random number generators, normal distributions to simulate, for example, what was the mean age, what's the mean proliferation Ki67 index, all based off of, obtained from that meta-analysis of the scientific literature that we did. And then we just pulled the patients together, these modeled patients, simulate fictitious patients. And we, and in this case, what I also want to mention, we didn't include any data on incidence. So essentially, in this simulation, every tumor entity is equally represented, more or less. And this is kind of an example of the data we get. You can see everything's a normal distribution. Here, we're looking at a particular type of disease called Anaplastic Astrocytoma, which occurs kind of in around 50, 55 years old. It has this type of proliferation index, and there's some other features of things that I've mentioned before. Just another example of 2 things. These are 2 types of tumors, both of which occur in the same distribution in the posterior fossa. That's a particular structure in the brain. One of them is an ependymoma, and one of them is a medulloblastoma over here. And you can see that they have very different proliferation indices, and they can be distinguished by certain features. So then what we did is we took all of our data here, as you can see, and we just performed a dimensionality reduction using the t-SNE model. And so in this t-SNE event, every single color represents a different brain tumor diagnosis that we had modeled. And when you just use clinical data, you can see that it's quite noisy and it looks like a disorganized plate of spaghetti and so forth. But when you add into the t-SNE analysis histological data, you actually start seeing the emergence of just visually some clustering. So if we're comparing this noisy relationship to this relationship here with clinical plus histology, we're starting to see very clear patterns. And if I add immunohistochemistry, you start seeing the emergence of even larger and easier recognizable clusters and so forth. To our surprise, when we added when we added molecular data, we didn't really see visually an improvement in clustering based on this. And so from this kind of thought experiment, we could conclude that upon visual inspection of the t-SNE, there's very little gain in clustering after you add data from immunohistochemistry. And so we wanted to think, well, what other things could we learn from a simulation like this? And for that, we turned to information theory, which I think that many of you may be familiar with, but I'll just define with a standard definition, which is that it's a mathematical representation of the conditions and parameters affecting the transmission and processing of information. This is routinely used in engineering and is the basis of many machine learning and AI workflows. I was introduced to information theory more as a basic neuroscientist. It's been implemented in neuroscience to understand circuits and from an electrophysiological plane, like which circuits are communicating and where the information is going in that context. So I was familiar with it from that literature. And so I was thinking, well, maybe we can think about that the biopsy is transmitting information to pathologists, and what does the pathologist need to get that data? And so in this experiment that we performed here, we took all of our data points and we just calculated the information that each parameter had. And we got some interesting results. So here in this graph, the x-axis is the information, and on the the x-axis there's the information. The y-axis are the different types of data points that are in the simulation. And three really important features predominate. So if you just look, the anatomical site of where the tumor occurs is the most important feature that has the most important information in this type of approach, followed by the Ki-67 index. That's an index of what is the proportion of cells that are in active cell cycle and dividing. Cancer cells divide, that's one of the problems. And of course, the patient's age. And so now you starting to understand, well, some of the challenges of implementing machine learning and AI in neuropathology is that when you couple this asymmetry of information, where it's basically and site proliferation and age are predominating and just really outperforming any other information you're getting, coupled with low sample volumes to train on, it'll be very difficult to achieve things. So if I were to lump all of the things together, here in this graph, I'm showing the purple line here is the full entropy in the system here. That's the measure of the disorder and how much information you need to achieve to get to your diagnosis. What's fascinating is in in this simulation, you have clinical information, just site, gender, and age, providing nearly half of the information you need. And histology and immunohistochemistry go almost all the way to getting you there. And this small difference that you can't even see on the graph, that's where this money problem is, okay? And so there's this practical challenge, which is that the molecular tests that are not helping us with the information, they provide significant prognostic information and treatment plan information. And so what's going on here is that different tumor classes share molecular features, and the prognostication varies as a function of tumor class. So as an example, there's a mutation in a gene called BRAF, it's a point mutation V600E. A BRAF mutation in a glioma would have a very different treatment plan than a BRAF mutation in a metastatic melanoma, okay? Completely different, okay? And so the tumor class and the mutation in the context of the tumor class, it was what matters. And so our approach to deal with this problem, and we're very practical, we try to find the easiest solution to anything, is to focus on a machine learning and AI workflow that solves a specific problem. So we find a specific thing, very granular, that we're trying to improve, and we think about different ways in which we can do that. So this brings me to this situation here, that we're going to talk about, which is the situation of diffusely infiltrating gliomas. This is a tumor entity that's commonly present in young adults. This class has two subclasses, astrocytoma and oligodendroglioma. The survival of these tumors is the astrocytoma has a mean survival of 7 years, whereas the oligodendroglioma can be almost like 14 years. The oligodendroglioma has some improved responses to different chemotherapeutics. So the oncologist actually has a bigger toolbox to play with the patients in an oligodendroglioma class. So one of the problems that happened in 2016, when the World Health Organization imposed molecular classification, is that to diagnose oligodendroglioma, it required documentation of something called 1p and 19q codeletion. These are chromosomal aberrations that need to be detected. This resulted in an expansion of the testing that for this particular entity. And so in one of the principles of pathology is when you expand testing and the incidence of a positive result in the population now goes down, the sensitivity and specificity all changes. And so there's three common methods to detect this. One is called FISH, or fluorescent in situ hybridization. This is fully reimbursable in the US, and in most countries, it's available in specific centers. There's two important aspects to this from a testing standpoint. It has about 5% of its cases are false positives. Well, given that prognosis of 7 versus 14 years, that's an unacceptable false positive rate. The good thing is that it has a very high negative predictive value. So if it's negative, it's, you're pretty certain about the result. There's another technique called LOH, which I'm not going to get into the details of it, but essentially it's essentially the gold standard. It's not reimbursed. And next-gen sequencing also can detect this. It's less commonly performed, but there's a whole series of reimbursement problems with that. So the specific granular question here is, when should we order this non-reimbursable test, which the hospital will just absorb and not bill the patient, when the 1p19q codeletion is positive on the FISH? That's our granular question. To approach that, we really had to start picking up not the phone these days, right, but the WhatsApp, and really try to get people from around the world to help us take a look at the epidemiology of this. And so we utilized some data sets such as the Cancer Genome Atlas, but we also got cases from all over the United States, from Galveston, Phoenix, Pittsburgh, put these together. We had colleagues in Argentina, in Brazil, and in Turkey, and essentially we were able to obtain epidemiological data of oligodendroglioma from a pretty broad data set. And so despite having that broad reach, we're only dealing with about 1,200 to 1,400 cases that we've achieved right now. Things that we can identify here are that astrocytoma and oligodendroglioma show significant overlap in terms of their age distribution. But as patients age, although the number of cases goes down, the probability of an oligo goes up. And that was very useful to us. Gender in this entity and this tumor seemed to not be particularly important. And so we received from these collaborators worldwide a structured data set. No slides. Many of them didn't have digital scanners, but we had just simple data. Age, gender, ATRX status, which is a biomarker, P53, which is a biomarker, and 1p19q codeletion status. So basically these here are binary readouts, and this is a continuous number. So we typically just generate dummy variables and performed a principal component analysis at first. And so here we're looking at the principal component analysis of our data set, our worldwide data set. I've plotted here the eigenvectors along principal component 1 and 2. And you can see some really interesting trends here. So here's our 1p19q status, and orthogonal to that is gender, which goes with the idea that gender is not really a driver in correlating with diagnosis. And then these two biomarkers, which are both utilized in most settings, they're extraordinarily redundant with each other, and they go in the opposite direction as the diagnosis. Here's some interaction with age. And if you apply information theory calculations, we applied this on 948 patients. You can see that the ATRX and P53 still has a lot of information to gain and to get to your diagnosis. So the idea was, can we use a simple imaging workflow to distinguish astrocytoma from oligodendroglioma? And we're focusing on simplicity here, coming from non-computational people. So we essentially looked at multiple tools that are available in open source packages and so forth. We created a ground truth by manually taking images of nuclei that were stained with a biomarker called Olig-2. And we tested basically 5 different techniques, 3 of which were unsupervised clustering, unsupervised segmentation techniques like deconvolution with OTSU, super simple, cut cluster classification, which is a method that was developed by a postdoc at the MBI here at OSU, k-means clustering, and then supervised techniques such as UNet, which is a convolutional neural network, and Weka segmentation. Long story short, when we evaluated the efficiency of segmentation, we found that as you increase the noise in the image, surprisingly, the deconvolution with OTSU performed just as well, if not slightly better than a UNet. and it was faster. These are some of the processing time here on the x-axis for the different techniques, and we can see that UNet has a much higher requirement than deconvolution. And so we basically stuck with this technique here. And so we took our slide, we took tiles of these points, we obtained nuclei, and we had a lot of segmented nuclei to look at. We have images that are, each nucleus has about 80 different features between shape features, intensity features, and texture features. And we essentially were really lucky to find so much redundancy in the features we had. We were able to reduce dimensions in the data set by going from 213 features to 8 features by passing this through a principal component analysis. And so one of the things here is, okay, so now we've got this. So what do we do with all this data, right? So one of the histological features that distinguish an astrocytoma from an oligodendroglioma is that the astrocytoma tends to have a much wider variance in the morphologies. The oligodendrogliomas tend to be much more monomorphic. And so we thought, well, this would be really simple then to model. Maybe if we were to plot this off onto a multidimensional space, if things are monomorphic, they will cluster more closely together than if things are much more pleomorphic or more variable. And so we decided to look at the density of the data in that multidimensional space. So we're not talking about density as in cells per volume, but rather density in that space. So as an example, if this is an example of a principal component plot, we're finding the 10th nearest neighbor across in terms of Euclidean distance across that multidimensional space, et cetera. And then we recalculated our eigenvector plot here to look at in this particular data set. Again, here's 1p19q. It goes on the opposite side of p53 and ATRX. These two are highly variable. And the features from the OLIC2 stained sections, which are what you saw a segment, there's some, they at least go to the same quadrant as a 1p19q. And so we applied an information theory approach on this here. We calculated the information gain. And this feature right here, which again is a super cheap feature to identify, compared to P53 and ATRX, it doesn't provide everything, but it certainly provides some information, and some information that was missing, that we could have extracted that we weren't extracting from the data. And so as I move towards the concluding remarks, our goal again was to identify when we have a positive, when do we order a fish? When do we order that expensive non-reimbursed test? So what we did, we generated a simple classification system using a random forest where we had diagnosis. Again, that's astro versus oligo as a function of ATRX, P53 status, the age, and these OLIC2 features. And we looked at these particular cases. So we have a total of 5 cases here that these 3 were actually false positive. These are ones where the fish was positive, and then the patient either recurred within a year or 2, which is not what an oligodendroglioma should do, or we'd had a reflex positive test sent out that the hospital just absorbed, and that proved that this was a false positive. And on the x-axis, what you're looking at is the probability of determined by the random forest votes in predicting if the case was 1p19q codeletion positive. Okay, and so you can see that this was really helpful in showing that in fact these 3 cases, despite a positive fish, had a low probability based on our algorithm to actually be positive. These cases here were essentially, you can think of as true positives. They were positive by fish, so it's kind of think of them as controls in terms of how well it's predicting true positives. So this leads to some certain clinical guidelines. So for example, ways that you can envision this is that if you look at these histology and you find that under this particular threshold, say a 0.3, you wouldn't even have to order a fish result. Boom, just don't order it. The probability of it being fish positive is very low. Likewise, here, if you're above a particular threshold, you would not order this non-reimbursable test. If you're in that middle zone, you would be in a situation where you would order this non-reimbursable test when the fish is positive. So again, this has a high negative predictive value. So if it's negative, we're fine, we can stop. But if it's positive, maybe that's when we should go ahead and do our testing. With that, I'll just stop to take any additional questions. I wanted to thank my team who did all of this work. The OLIG2 modeling was done by Dr. Sevic. The glioma simulation was worked on by Dr. Aslan. Dr. Kamat, who was a Fulbright scholar in my group, but had to leave early because of COVID-19. Dr. Garragorri, who's a pathologist from Uruguay, who spent some time with us here. Professor Thomas, who helped us with a lot of the intellectual clinical components of this. And Dr. Vasquez, who's the mathematician who developed the cluster classify method, and Wesley Wang, who's one of our grad students. And the collaborators really all over the world that helped us to generate this data set. And with that, I'll take some questions.
So thanks, Jose. So I think there have been several questions on the chat. Does anybody want to read those out, or should I do that? People can directly ask questions.
I have a verbal question. It wasn't in the chat.
Yeah, sure, Ross, please proceed.
Yeah, so first of all, Dr. Otero, this is a really interesting talk. Thank you for this. I think this kind of view of this as being, from an information theory point of view and from a statistical point of view is really interesting. And I was also very interested in this kind of, how should I say it, synthetic or simulated data set. And I was kind of just thinking to myself, like, sort of how far someone could push that. So, if I understand correctly, you sort of invented patients with demographics and diagnoses, but then you also added histology images. Is that true, or did you just add histology outcomes to that?
So let's think about let's think of histology here, histological features as a biomarker. Let's just put it that way. So. So the way that we generated this is one histological feature that's important is something called necrosis, for example. So let's say necrosis is the biomarker. The way we modeled it, it's just super simple. We said, okay, we're going to randomly sample zeros and ones, one being necrosis positive, 0 being necrosis absent. And we're going to, based on the scientific literature, we know that, let's say, for example, polycystic astrocytoma, that does not have necrosis. We know that high-grade gliomas, like for example, Senator McCain died of, about 80% have necrosis.
Then you generated these biomarkers in a sort of statistically sound way that reflects the actual.
Yes, it was based off of the meta-analysis that we had done of the scientific literature. So we have, and the WHO, they have, it's like 50 or so tumor classes, and there's descriptions between those, and of course the histological features, they overlap, but things like anatomical site, they tend to change, and so forth. So that's how we simulated that in that context. So it was a pretty, it took a lot of time to get the meta-analysis. Francisco Garragorri from Uruguay is the one that helped us with that when he was here. But it's actually like a quite simple workflow to actually do it.
Very interesting. Thank you.
So might be just for the sake of time, we can move ahead so that we don't lose time. And Dr. Otero, again, thanks. There are some, please follow up with the chat windows. If there are any other questions, you can answer them.
I'll just do the stop share now, right?
Okay. Yeah, thank you.